Well, good evening, everybody, and welcome to another in our inaugural series of lectures. It's a great privilege and pleasure to welcome Professor Jeremy Turnbull, who's giving, if he's to be really honest, this is his second inaugural lecture, because I know he gave one at Liverpool when he joined Liverpool University as a chair, and I was at that university at the time, although I missed that lecture, so I'm able to catch up since. Just a little bit of background before we get going. Jeremy is a professor of glycoscience at Keele University, and we were lucky enough to recruit him a few years ago from Liverpool. And his expertise is in proteoglycans and extracellular matrix and areas in glycomics. I'm not going to pretend to go, and certainly won't do justice to the depth of his research. I think he's going to tell us about that coming forward in a moment. But just to say, he started his career, he moved to the UK for his education, was educated at Swansea, and then PhD at Manchester University in 1990, and then in 1996 he was rewarded a senior research fellowship by the Medical Research Council, and he started his own lab at Birmingham University, and he had that fellowship renewed in 2001, 2003, very prestigious set of fellowships there. He then moved to the University of Liverpool, where we shared the same employee for quite a few years before I moved here to Keele. Before his education, Jerry was actually born in New Zealand to Australian parents and brought up in Sydney and Brisbane in Australia. And he came to Europe, as I mentioned, as a teenager. He spent a year travelling around Europe in a camper van with his parents and brothers, and eventually settled in what's now known as Happy Valley in West Yorkshire, Hebden Bridge. And that's apparently before it was trendy, or maybe you made it trendy, I'm not sure. He studied for his local, his A-levels in a local comprehensive, and where he studied, noted here that he had excellent biology and chemistry teachers, and I think probably his lecture is going to be a testament to the inspiration that those early teachers gave him in those subjects. Very importantly, Jerry still supports Antipodean teams in both rugby and cricket. However, the circle of life has turned, according to his notes, and his son has recently married a Kiwi and now lives in Auckland but supports England. So that must be quite an interesting conversation on the Boxing Day test, or the equivalent when you all meet up as a family. So without further ado, I'd like to welcome Jerry to give your lecture this evening. The lecture is being streamed live online. I think it's actually going out to Australia and New Zealand, although whether anybody's awake at this time to watch it, we're going to watch it in catch-up, I don't know. So welcome very much to our online audience. And at the end of the lecture, we'll have a chance for a few moments of questions. So thank you very much, Jerry.
Thank you, Jonathan, for that really nice introduction. And it's a great honour and a great pleasure to have the chance to give an inaugural lecture, in fact a second one, as was pointed out. A nice opportunity to reflect on what you've done in your career, and looking forward as well. A bit less time to look forward this time than last time, but still looking forward. So what I think I will tell you about a little bit is initially set the scene, tell you a little bit about the molecules that I'm interested in, sugars, and the study of sugars, glycobiology, and the particular type, the proteoglycans, that I've been interested in throughout my career. Tell you a bit about how I got into this whole area, and then give you some examples of some flavour of the different research that's been going on, past and present, in the various places that I've worked, and then obviously finish with acknowledgements. So glycobiology is basically the study of sugars, particularly sugars attached to proteins and lipids, where they have really important functions. And then glycoscience, which is the name of the new centre here, which it's great that Keele has been able to recognise the research strengths in that topic here and create a centre, and it's great for me to be involved in that. That's essentially the broader field of glycobiology, where it meets chemistry, materials, biomaterials, medicine, etc. So glycoscience is kind of all-encompassing. And I guess it's really arguable that it's still pretty much a frontier field, still at a relatively early stage. and there's still a lot more to be done. And the key thing about sugars is, unlike DNA and protein that are the template-driven, the synthesis is template-driven, so DNA is copied to RNA, which is then interpreted by the cell machinery to make proteins. Sugars structures are not encoded in that way. They have to be made with enzymes, so they're not encoded directly by DNA sequences, which makes it a little bit more tricky to work on. And in fact, techniques largely have lagged behind in this field, and many people working in this field actually find they end up having to be technical and develop methods as well as actually using them to study things. And certainly that's my experience. So I guess for me, sugars, when I look back, go back a long way, because my A-level research project, which were the Nuffield research projects we did then, was studying salivary amylase and its digestion of starch in the mouth. So I started getting interested in sugars already at that stage. And sugars are really important because they modulate the activity of many different molecules. And traditionally, of course, you think about sugars, energy sources, structural molecules like plant cellulose, etc. But sugars themselves have an incredible structural diversity to them, which then means they have an amazing amount of different specialized functions. And they're essentially very information-rich molecules which biology is using in all sorts of really interesting and exciting ways. And so you have monosaccharide units of various different types linked together in all sorts of patterns and shapes. And, for example, the blood group antigens are made up of particular combinations of sugars. So that's what encodes the different blood group antigens that we all have as individuals. That's based on sugars. But they also modulate protein activity. So they're not just there as a kind of structural component or necessarily just for recognition, but in fact, most proteins are actually glycosylated, especially those that are outside the cell. Almost all of them have sugars on them. And those sugars can modulate the protein activity. They can also have their own independent activities. They can mediate the ability of that protein to be recognized by other proteins, which is really important in biology. But they can also modulate the protein activities. And essentially, they've become something that's developed as organisms have become more complex through evolution. In order to have multicellular complex organisms, you have to have sugars, an elaboration of more and more complex sugars in order to encode much more complicated activities than you could do with proteins alone, for example. So in fact, the surface of the cell is actually decorated very heavily with sugars. So in fact, most proteins on the cell surface, either embedded in the cell surface or attached to the cell surface, actually have sugars of various types on them. And some of these have very long chains, and these chains called glycosaminoglycans, which are the ones that I've worked on, attached to proteins, are called proteoglycans, and we'll talk a little bit more about those in a minute. But if you look at an actual picture of a cell coat, you can see it's kind of quite hairy, and it's because of all these glycosylated sugar-attached proteins on the cell surface. So I guess the second area where I continued with sugars after I got my biochemistry degree was actually working at Christie Hospital in Manchester and working on breast cancer antigens, and those turned out to be glycoproteins as well. And in fact, my first paper that I published was on breast cancer antigens, and that was working with Professor Tony Howell. And I was actually employed as a hospital biochemist, so I wasn't doing a PhD initially when I started after university. And then down the corridor, there's this interesting chap called John Gallagher, and he said to me one day, well, what about doing a PhD part-time on proteoglycans? And I thought, that's interesting, but what are proteoglycans? And I dug back through my lecture notes, and I think I found one lecture which mentioned glycosaminoglycans and proteoglycans, so I had to find out what they were and dig into it a bit more. And essentially, I ended up working with John and doing my PhD with him on these molecules, heparan sulfate proteoglycans. So, like the picture I showed you earlier, these are molecules attached to proteins, either on the cell surface or outside the cell, and they have these long chains of sugars, repeating sugar units, which are decorated with a very interesting chemical group, sulfate groups, which makes them very negatively charged and able to stick to basic patches on, positively charged patches on protein surfaces. So they're really interesting in the way they interact with proteins. They're really important in the matrix for outside the cells as a whole mix of proteins that allow cells to stick together and create the shape around cells. And proteoglycans are really important as part of that. And I was particularly interested in this class of molecules, which John Gallagher was a world expert on, and I was very lucky to have the opportunity to work with him, and it ended up being what I've worked on my whole career. Not that that's always what happens, but it did in my case. So these molecules have turned out increasingly to be very interesting because they have all sorts of functions. They're sitting on the cell surface, so they're in a very strategic location where they can interact with lots of molecules, interact between cells, etc. So they seem to be important in things like cell-cell adhesion, presenting chemokines and growth factors, which are proteins which cells use to communicate with each other and elicit responses, clustering and signaling of receptors, the matrix, as I mentioned, depots of molecules in the matrix, uptake of molecules by endocytosis, all sorts of interesting functions. And they do this through these interesting structures. So this repeating disaccharide unit that can be decorated in different positions on the sugar rings with sulfate groups in the position shown here with the red symbols of the different positions on the sugar rings. And so this is a really interesting area of chemical space within biology because you can create an immense number of different types of combinations of those structures as the sequences get longer. And the basic idea is that these molecules are then selectively interacting with different proteins and then thereby controlling their activities. So it's a little bit like a dimmer switch on a light, not so much an on-off switch, but rather, depending on the range of structures that are present in a particular cell at a particular time, that can influence its ability to regulate different proteins. So it's a kind of ensemble of different bioactive structures can then tune the functions of those cells. So when I started my PhD, there really wasn't a lot understood. There was kind of a lot of interesting anecdotal evidence for interesting functions for heparan sulfate, but not a lot was known. So really there was very little in the way of methods for studying structure and function. So I guess what I originally started out doing was to try and think about changes in cancer, although we never really finished doing that because it ended up being a lot of work to try and work out what is the basic structural order and complexity in this molecule. And that required development of novel methods, as I mentioned earlier. And of course, this is a nice quote from George Orwell. In order to actually, you're limited by your technical developments, basically. So it's an essential thing to develop new methods. And I had some nice opportunities on the way. During my PhD, I had the chance to go and work at the University of Iowa with Bob Linhart, who's one of the experts in the field on structural analysis of heparin and heparan sulfate. Many of you will have heard of heparin, which is a very widely used anticoagulant drug. And that's actually a member of the heparan sulfate family as well. And we worked together with him on some of the enzymes that certain bacteria in the soil produce to break down heparan sulfate. So they're basically soil bacteria. They're designed to break down tissues of animals that die and degrade in the soil. And we were able to work out some interesting features of how these enzymes cleave at different positions depending on the sulfation patterns. And that allowed us to then use that to understand the structure of heparan sulfate better. But it's a great opportunity to go and work in his lab for a couple of months. So essentially my PhD ended up then focusing on trying to understand heparan sulfate structure, developing methods to enable us to do that, discovering what the structural order was and how complex it was. And one of the things we did was develop a gel mapping method where we used gels such as those that are used for separating proteins and DNA and could do some nice high-resolution separation of different oligosaccharides after the chains had been chopped up into fragments. And that immediately told us when we compared different cell types that these things were really complicated. Look how many structures there are present when you chop this up with a particular enzyme. It's really quite complicated. And that also led to development, as I'll mention in a minute, methods to kind of work out what the sequence of the sugars was, what the order was. And this was actually one of the first really important conferences I went to, the Gordon Conference on proteoglycans in 1988, and I presented that oligosaccharide mapping method. And if you're eagle-eyed, you can see I'm up here in the top. This is John Gallagher just here as well, and Bob Linhart. So it was a fantastic, in the kind of middle stages of my PhD to be able to go to this kind of prestigious conference and present data was very inspiring. And I've been going to that conference almost every 2 years since, and in fact became the chair of that conference in 2016, which was also a great honour. And we had a lot of fun as well. This is the famous Ironman competition that was developed by another Aussie, Bruce Caterson, emeritus professor from Cardiff, now retired. He got this going some years back, this was the third Gordon conference, so I think it started with the first or the second one. And you can see I'm just here actually getting ready for my first Ironman competition at the back here. A really fun race. There's a duck pond in the conference site, and basically you have to have a drink, swim across the pond, run, eat a Twinkie, which is a horrible American cake, and then run back and tag the next team member, and there's 4 team members, so it's quite entertaining, and it happens at 6 o'clock in the morning on the last morning of the conference, so after the party the night before, so it's pretty challenging. So plenty of fun along the way as well. So I guess this slide in a way summarizes some of the key things that came out of my PhD basically, which was discovering really that these long heparan sulfate chains were actually quite ordered. Although there's quite a lot of complexity in terms of the individual oligosaccharides, they're arranged in the chain in a way that the sulfated domains are spaced apart. So you have these N-sulfated domains where a lot of the additional sulfation happens in red. These blue domains are essentially unmodified spacer sequences. And then you have these intermediate N-acetylated, N-sulfated domains sort of mostly in between the two. And that, pleased to say, that model has stood the test of time and is still current and applicable. So you get variations in that exact model depending on which cell or tissue you look at heparan sulfate from. It's a dynamic system and it's variant with different cells and tissues, but that basic structure is proved to be correct in the case, and obviously suggests interesting thoughts about how this molecule might function and space protein complexes apart and so on. Still a lot of work going on that. But I thought I'd show this slide because this is, I think, from the first poster I did on this work, which was probably about 1990, just to remind people that we didn't have, well, barely had computers when I wrote my PhD in 1990. We had BBC Acorn computer or something, or what it was, I think, to do it on 5¼ inch floppy disks. But we had to draw our own structures and do stenciling and various things to do posters. So quite a lot of effort to do that. But this was one of the early models that we drew and presented at a conference. And this was actually, my kids used to come and visit the lab, of course, and my daughter Sophie was obviously inspired by things she saw in the lab and drew this picture, which I thought was actually a beautiful picture of a proteoglycan, because you've got the core protein here, and then you've got these chains radiating out with these very different sequences and patterns along the chain that are important for interacting with different proteins. So that's one of my favorite pictures that she drew. So one of my favorite cartoonists, Michael Leunig, an Australian, did this cartoon, which I really liked. Someone going to a guru in a cave, and the guru saying, well, I wouldn't know, I'm hiding in a cave, basically. I haven't read the latest research yet. So for me, I guess the happiness ended up being working on heparan sulfates, and that's certainly been a great joy and a really interesting field to work on. So the big question, which we're still grappling with, but it's certainly been something that started in my mind at that time, was, is the sequence of the sugar units in heparan sulfate important? As Shakespeare says here, everything has to be in order in the heaven and the planets and so on. So we're really still interested in that issue. But at the time, one of the approaches we developed was to use some of the natural enzymes that break down the molecule to get information about the structure. And I had the chance to go and work with John Hopwood in Adelaide. He was working on lysosomal diseases. So these enzymes are present in the lysosomes, which break molecules down inside the cell. And there are children that have defects in the enzymes in this pathway and have all sorts of problems with skeletal abnormalities, neural abnormalities, and so on. And his lab and a number of others have developed now successful enzyme replacement therapies and gene therapies now coming into play as well. So at that time, they weren't finished, the development wasn't finished, but now they have been. So they're basically studying the enzymes that work on these oligosaccharides from one end, so they only work at one end, and they clip off different groups specifically from the end in the reverse of the biosynthesis, basically, the way the molecules are built up. So we figured that if we used these enzymes to see which groups they clipped, whether they clipped off a group or not, would tell you whether that group was there. And we married that up with our gel-based methodology. And this shows one example where we're sequencing an oligosaccharide. So you basically put a fluorescent marker on one end of the oligosaccharide, and then from the other end you clip away with either a chemical treatment or the different enzymes, and depending on whether you get a shift tells you whether that group is there or not. And basically it's a gel shift type methodology, and we were able to publish that work, and that was one of the methods that we had available to us to start working on at the time. Now we're using mass spectrometry to do basically the similar thing, but with much higher resolution. And in the spirit of this quote from John Nass, the famous mathematician, film Beautiful Mind, if you've seen it. Never be afraid to try something new. Remember, amateurs built the Ark, professionals built the Titanic. I went and took up my a fellowship and started my own lab, as Jonathan mentioned, in Birmingham, and that was in 1996. This picture was probably taken a few years after that as the lab developed, because initially, obviously, I didn't have anybody, and then it sort of gradually developed as I recruited people and got grants, etc., and took on PhD students. But over time, developed a really nice group there. And we got interested in a whole range of topics that were coming out of some of the things I've just been telling you about. Really, how does heparan sulfate structure vary, and how does it interact with proteins? We made libraries of different natural structures and also chemical analogues of them. We wanted to understand how the dynamics of heparan sulfate biosynthesis and how it functions in different systems like the mouse brain and stem cell differentiation. Got interested in neurodegenerative diseases, and I'll tell you a bit more about that in a moment, because that's one of the directions we went in. And also studied proteoglycans in the worm C. elegans. So using a whole range of techniques, biochemistry, cell biology, chemistry, molecular biology, and model systems. So it was a very exciting time. Good timing in terms of being able to get funding. It was the Blair government and a lot of investment in science. So for me, it was a little bit of a sweet spot to be coming in and trying to get started at that time. Very fortunate to have had that opportunity. So I guess one of the key things was really to try and understand how heparan sulfate regulates protein function and what the specific sequences are that interact with different proteins, what mechanisms do they work by. And we knew from a number of studies that they work through a range of different different kind of mechanisms, the most famous one being affecting the conformation of the protein when they bind to it. And this is how heparin, which binds to antithrombin, actually alters its conformation and activates its ability to inactivate factor 10A in the coagulation cascade, and that's how heparin works as an anticoagulant. So that's probably the most famous mechanism. But increasingly, as time went on, it was discovered that also it can bring proteins together, binding together along the chain, sort of bringing them into close proximity. But then, very importantly, working as a co-receptor with extracellular ligands, so bringing growth factors, which are important for communicating between cells and instructing cells what to do, bringing them together with their receptors and giving you productive signaling, and that's something we got really interested in. And also working as a cell surface receptor to bind molecules and take them up into cells as well. So it was a really exciting time to be working in the field, because all these questions were open and there was beginning to be the tools to really study them. And so we did quite a bit of work on one signaling system, the fibroblast growth factors, which they're not just involved in stimulating fibroblasts. They are involved in all sorts of different cell systems, in cancer, in stem cell differentiation, and so on. And it had been found that heparan sulfate was an essential co-receptor to bring those signaling complexes together. And we were able to show the first binding sequence for a particular FGF, FGF2. This is work I did with John Gallagher in my early postdoc years. And then we also looked at, when I started my own lab, at ligand and receptor specific signaling. So this was work with Scott Gimmer, who's actually in the audience and is one of the people from the lab that stayed with me for quite a number of years, my first postdoc. And we made a library of natural heparan sulfate structures, which was very complex, and screened it with different FGF ligands and looking at signaling with different cells with different receptors in them, and looked at how the responses of the cells differed. And we were able to show that it depended on the structure of the heparan sulfate oligosaccharide, the particular ligand, and the particular receptor. So there seemed to be quite a bit of specificity to it. And we also had indications that it was probably to do with the kinetics of how long you keep that complex together, made an important impact on the cell signaling responses. So then in 2003, there was an interesting occurrence, which the sporting interest has already been alluded to, and 2003 was not a good year for Australians living in the UK. We lost the World Cup to Johnny Wilkinson drop kick in the dying seconds of the match, so that was a tough one to take. But for me, it was also a good year because I was offered the Johnston Chair of Biochemistry at the University of Liverpool and took that up. My senior fellowship had also been renewed recently as well, so that was again a great time for me to go on and develop my lab further. In fact, a number of people who'd been working with me in Birmingham actually moved with me to Liverpool as well. And if you might start to spot not only Scott, who's here, but also Mark, who was doing a PhD in the lab at the time, Mark Skidmore. And yeah, it's a really exciting time. And I guess we were, again, still really focused on trying to understand how heparan sulfate is made biosynthetically through a complex process involving a whole set of enzymes that elongate the chains, knit all those sugar units together, and then decorate them with different sulfate groups in particular positions. And really trying to understand how those addition of those sulfate groups makes these molecules more functional and gives selectivity to those functions. And so if you think about it a little bit like cupcakes, you can have a basic cupcake, but you can start to elaborate it by putting interesting toppings on it. And then for some of the structures like 3-O-sulfates and some 6-O-sulfates, it's like the cherry on the cake that really affects the activity and changes the activity of the molecule. It's a good way of thinking about it. But of course, what's the biological relevance? Well, heparan sulfate, as already mentioned, is cell and tissue specific. But we increasingly trying to understand how much variation there was at the cell and the tissue level that might encode different functions. And we also began to learn that the biosynthesis is both spatially, in space and time, dynamic. So different cells can change the structures they're making depending on the functions that they're undertaking. And maybe that reflects functional selectivity. The changes in structure affect the ability to alter the function of the cells and might tune multiple functions simultaneously. And just a couple of quick slides to tell you the sort of thing we were doing to get indications of that. We were studying neural development with collaborators at the University of Melbourne, and we were able to show that neural precursor cells in the developing brain, just a couple of days apart in development, completely change the chains that they're making. So they might start to make longer chains with more sulfated domains and an altered disposition of the sulfate groups on those sulfated domains as well. So really interesting changes in structure. And we knew that those could alter the signaling responses. So again, looking at FGF signaling between two different stages, we were able to show that the expression of the enzymes involved changes, that the structures change, and that the signaling responses to different ligand receptor combination changes in these cells. This was cells in vitro. But then we were also able to look, and this is where we never got enough data to publish this, but we looked at expression of one particular set of enzymes, the ones that put the 6-O-sulfate on. And if you look in the different stages of the developing cortex of the brain, the pattern expression of these three different enzymes is completely different in the different layers of the cortex where all these different types of neurons are being born and developed and elaborated. So really interesting in vivo data that these kind of changes in structure in heparan sulfate are underpinning potentially different important functions in the developing brain. And you can see that as well if you look with antibodies that bind to different heparan sulfate structures. You can see quite distinct patterns depending on which antibody you use, which suggests that those structures are varying in those different layers within the cortex. So more work to be done there. And the other area where we were got really interested in was stem cells, because stem cells are critically regulated by a range of different growth factors, such as the FGFs and others that I mentioned before. It's important for their self-renewal, their ability to keep propagating themselves, and also for their differentiation down specific pathways to become new tissues of different types. And so it's almost like, because a lot of those factors bind to heparan sulfate, it's kind of obvious that will be an interesting thing to study. And we did that in a number of systems. Some of the published work with Dada Pisconti and a PhD student, Rachel Gardiali, we were able to show interesting changes in heparan sulfate in muscle stem cells, which are really important in repair of muscle. When you damage your muscle, these stem cells get activated and they repair the damage. And we've done other work, and this is not yet published, but Rua Bhatti, who did a PhD with me, looking at breast cancer stem cells, we were able to show that if you treat those cells with different heparin and heparan sulfate structures, you can modulate their phenotype. You can make them less like cancer stem cells. And you can also enhance their sensitivity to chemotherapy drugs as well. So some interesting ideas that we might be able to maybe target these kind of cells, which are really critical because these are the cells which seem to be normally resistant to chemotherapy, and then after treatment, the tumor will regrow from those cancer stem cells. So maybe it's a possibility to create some adjuvant heparin treatments to make cells more sensitive to chemotherapy treatments there. But I think, I guess, increasingly as my research went on, it got kind of pushed more and more into, well, what are these molecules doing and how can we use those in a biomedical sense? And of course, glycotherapeutics have actually been around for many years. vaccines. A number of vaccines are critically involved. Polysaccharides, for example, blood group antigens I've mentioned. But heparin itself, which is the anticoagulant drug, was discovered originally in the sort of early 1900s and went into clinical use around 1930, and has gone through a number of generations, improvements with low molecular weight heparins, and then ultimately a fully synthetic chemical structure as well, a mixture. This 5-mer sugar, this 5-sugar unit structure, which can be synthesized chemically. So this kind of gives you the idea that there's potential to develop these kind of molecules for other applications. And as time's gone on, and a number of structures have been, or a number of routes to make compounds which you could use potentially as therapeutics have come forward. We use natural product libraries for a lot of our discovery research, but we started developing modified heparins where you could play tunes with the sulfation patterns through selective desulfation chemically of heparin molecules. So this is engineered semi-synthetic heparins, which don't have a coagulant activity. And then we've collaborated with groups making fully synthetic heparan sulfate structures. Very difficult chemistry to do that. To make them using enzymes as well is something that groups have begun to develop. And also we've collaborated with groups making glycomimetics, where you're making something that mimics the natural structure but is much easier to synthesize. So this is a kind of a simple, well, it doesn't look that simple, but a fairly simple multi-armed chemical structure. And then putting some sulfated sugars on the end is relatively easy compared to making a long, fully natural structure. So these have proved to be very active molecules as well. So we've got a nice suite of molecules to test in different systems. And we started doing that in different applications, and I'll mention some of those now. But basically the idea is that proteoglycans, because they're very active in a range of different biologies related to different diseases, that creates a rich territory for drug discovery. You can target molecules towards the proteins that they interact with involved in those functions. And you can also target inhibitors of enzymes related to heparan sulfate. So there are enzymes on the outside of cells, this enzyme here, heparanase, which clips the chain into fragments, and that turns out to be important in aspects like cancer and other biological systems, nerve repair. And then sulfs, which are these enzymes that specifically remove the 6-O-sulfate groups from heparan sulfate chains. So they're another potential target. Because they're outside the cell, they're relatively easy to hit. So we have some interesting tractable options to make compounds, and what we tend to describe these now are as next generation of heparins for different applications. And I'll just run through a few slides on some of the topics we've been studying. We've been looking at inhibitors for Alzheimer's disease, COVID therapeutics in the last few years with the pandemic, studies on nerve repair and multiple sclerosis, and also some inhibitors of cancer cells. So we got interested, I guess, originally back in 2003 in Alzheimer's disease, because it was known that heparan sulfate proteoglycans are found in the amyloid plaques, where amyloid peptides, which are one of the key known causes of Alzheimer's disease, actually accumulate outside the cell. And that's what got us interested initially. But then it turned out that one of the key enzymes, which is here, which cleaves a longer protein sitting in the membrane, the Alzheimer precursor protein, it clips it, and another enzyme clips here, presenilin, to make these fragments. And it's these are the peptides that accumulate in the brain of Alzheimer's patients, and are one of the first causes of the cascade of problems that cause the ultimate disease. So it's a key enzyme. This enzyme's a key enzyme because it's at the first stage of making that peptide. If you can inhibit that enzyme, you can stop the whole cascade happening. And so it became a big target for pharma. And because we were studying the plaques, we were working with GSK at the time, we actually identified that heparan sulfate is the natural inhibitor of the beta secretase. It depends on the sulfation and the size of the oligosaccharides. And that led us to ideas about maybe we can make a strategy for countering excess base activity and reduce amyloid levels in vivo. But we also went on to do further studies, Hannah Clark for a PhD, looking at whether heparan sulfate might underpin the natural regulation of amyloid in vivo, and we have some interesting evidence for that. So there are associations between genes in the heparan sulfate biosynthetic pathway. We also are able to purify heparan sulfate from the brain of Alzheimer's patients and show that it had reduced levels and a lower level of sulfation, which was less able to inhibit the base. So the idea was it would release the brake on, might be a natural brake on base, and that if you don't have the right structure, that brake is released and you produce a lot more amyloid. So this could be a mechanism contributing to elevated levels of amyloid. And genes related to heparan sulfate might be novel modifiers of susceptibility. So I think that's really interesting, but we'd already thought about the idea of maybe trying to make drugs to counter this issue naturally. And the first way we did that was work of Ed Yates, who's a long-term collaborator, started in my lab in Birmingham, is now working in Liverpool in his own lab, generating libraries of modified heparins where you chemically alter the sulfation patterns, and they give you a nice lead into what structures are important for what activities, what sulfation patterns. And we were able to identify some interesting compounds which essentially we'd knocked out their anticoagulant activity, so they'd have no side effects like bleeding, but they still maintained their ability to inhibit B secretase. We were able to show that they had sort of favorable properties, no toxicity in animals, low off-target effects on other proteins. And we're also able to do, this is some nice work that Scott developed, a nice cortical brain slice assay where you could take slices of brains from mice that develop amyloid very rapidly, transgenic mouse, and look ex vivo, so growing it in a dish, basically. You could look at the ability to inhibit the amount of amyloid produced by that tissue. And we did some initial work with Jinping Li at Uppsala University, who had a mouse model of Alzheimer's, and we're able to show that we treat them with one of these compounds, an N-acetylated low molecular weight heparin. For a month, we could reduce the amount of amyloid in the brains of those animals, consistent with the expected mechanism of action inhibiting base. So that was an exciting start point. In the meantime, we'd also thought about the idea of making chemically produced structures that could target the particular inhibition of base. And so we knew from the modified heparins what kind of structure to go for. We hooked up with some fantastic chemists at the University of Wellington in New Zealand. You can see a pattern here, collaborating with antipodeans, really useful for traveling over there. And they made a really exciting library of a range of structures from hexasaccharides up to, in fact, dodecasaccharides, so 12 mesaccharides, with a range of different structures consistent with what we wanted. And we were able to screen those, and we actually identified some that had really quite nice potency that made them drug-like. Of course, an important question is, can you deliver those orally? And because for Alzheimer's disease, you really want an oral drug. You don't want to have to give injections. And we were able to show that a number of those compounds could actually get access to the brain through oral delivery. So we radiolabeled them, gave it by oral gavage to the animals, and we're able to see significant levels building up in the brain, and other tissues as well, but certainly in the brain. And we were also nicely able to show in some preliminary studies treating just in the drinking water. So we just put the compound at the right level in the drinking water, knowing how much water the animals drink to give them a targeted dose, and that we were able to show a reduction in amyloid with an oral treatment for a month. So we then targeted a couple of structures where they could scale up to make enough to do proper, sort of more significant animal model studies. And we picked, two octosaccharides, which are a little bit easier to synthesize than the longer saccharides, but they still had good potency. Two different octosaccharides that have different levels of sulfation. And they were able to scale those up to a level, sort of almost to gram level, certainly hundreds of milligrams, in order that we could do a long treatment in the mice. And this is a study that's taken quite a bit of time to develop because the chemistry is very difficult and the scale-up is all new, and this is funded by the Alzheimer's Society. And this work's been going on over the last year after the making of the compounds, and obviously all the problems with COVID and so on. Our collaborators at Shanxi Medical University in China were able to do some really nice studies where we treated the animals for 4 months, starting at a relatively young age. So this is a transgenic model where they have 3 different mutations known to cause Alzheimer's disease, so they get very early development of Alzheimer's disease, so you can keep the mice for a relatively short time, see whether your treatment's having an effect. And we looked at biochemical measures of the disease, behavioral studies to see whether we were affecting things like memory, and also they looked at proteins related to synaptic plasticity, so whether the brain was succumbing to damage to the synapses and communication between the neurons. And the basic idea of the treatment was to go in relatively early in the cascade. So one of the issues that's coming out in Alzheimer's is that by the time you see symptoms, a lot of the amyloid cascade has probably already happened, and that's probably why a lot of the treatments haven't worked yet, because they haven't been given early enough. Later in the cascade, you get problems with tau, and then obviously you get cognitive decline happening. So this is a kind of series of cascades of events that happen sequentially. So if you could go in here a few years earlier and treat, the idea is that you might be able to stop this cascade, stop these levels building up to the level where they precipitate these subsequent events, and maybe you could actually stop the disease in its tracks. So so-called prodromal treatment. And I'll just quickly go through these slides. So we were able to show very nicely in these studies that the level of amyloid proteins was significantly reduced. So here's the normal animal, the so-called wild-type animal, relatively low levels of amyloid. In the transgenic animal, those levels go up dramatically. And with these treatments, we could push the levels down much lower. This is amyloid beta 40, and this is 42. So they're back down to almost near normal levels, which is really encouraging. And then in a number of tests, behavioral tests, are able to show significant rescue. So here, the wild type shows a deficit in short-term memory, so a reduction here. And then when we treat with these two different compounds, we can elevate and improve their short-term memory back towards the control, the normal level. In a different test of spatial memory, we see something similar, although compound 17 looked a bit more effective. That pretty much completely rescued the deficit in the transgenic animals, and compound 16 was a little bit weaker. So this is a water maze test where the animals have showing a recognition or remembering where to go when they're swimming in a maze. And then a different kind of measurement, a so-called water escape task, where they have to find their way through the water and get onto a platform to get out of the water. And over time, you can see the wild-type mice, the normal mice, they learn, and the amount of time taken to escape goes down because they're learning. In the transgenic animals, they learn a little bit, but then don't really learn anymore. They perform very poorly. And in these animals that with the treatment, you can see considerable rescue of that activity back down to sort of near normal levels. So these are really quite exciting results, and also in terms of synaptic proteins. So these are proteins which give some indication of the status of the synapses and the connection between neurons and the activity. And you can see again in the wild type, there's a certain level of these two different proteins, and there's a significant deficit in the transgenic animal. And with our compound treatments, we can rescue quite significantly back towards, or very close to, in some cases, to the normal level. So very exciting that there's probably some improvement in synaptic plasticity, and that would be consistent with what we saw in the behavioral experiments. So I think very, very exciting data in this system. It's one that we've been studying now for quite a long time. And we have a number of compounds that have these kind of effects. They're very, very sort of encouraging properties, potentially favorable for preclinical development. We can give them orally, for example. They're potent, etc. They don't show any toxicity problems in what we've seen so far. So we think that these kind of molecules or other molecules of a similar type potentially could be very exciting to rescue the levels of, or lower the levels of amyloid beta long-term treatment if they can be given early enough, maybe with better biomarkers and the ability to define the disease onset earlier. These kind of drugs could be very useful. So potential long-term safe treatment to modify the disease progression. And it's encouraging that recent antibody trials have shown some improved efficacy of anti-amyloid therapies, but these are obviously very expensive and also have significant side effects as well. So I think if we can come up with something that's based more on natural compounds, it would be potentially very exciting. I'll just have a few slides then covering a couple of the other topics. So COVID, of course, has influenced all our lives. And it turned out that heparan sulfate's important in that as well. And in fact, the group here at Keele, working with them when I was still at Liverpool initially at this point, were the first group to show that heparin binds to the SARS-CoV protein very early in the outbreak. We published a preprint in March 2020 that led to a lot of interest in heparin as a potential treatment, and further studies around the world, so that's very exciting. But we're able to show that heparin and related compounds actually bind directly to the spike protein on the surface of COVID, and also interfere with the binding of the spike protein to the ACE2 receptor on human cells. So, meaning that there's potential to use those molecules based on that to interfere with COVID infection. We collaborated with Public Health England early on as well to look at different heparins and their ability to inhibit binding, and the take-home messages from that were that normal unfractionated heparin probably would be suboptimal systemically given to try and treat COVID. The peak dose level when you give heparin was probably just around about the IC50 for inhibiting the virus, but more likely could be useful for direct lung delivery. And indeed, there have been a number of studies and ongoing studies to look at delivering heparin via nebulization to the lungs as a treatment for COVID, where you get much higher levels and you definitely get very good inhibition of the virus. Low molecular weight heparins, which are much shorter, were much less potent, which is very interesting and important to know. But we were also interested in looking at synthetic compounds because heparin, there are limited amounts of heparin available in the world, and if suddenly everyone wanted to take them for COVID or some other treatment, then there would be potentially a problem with the supply. So we collaborated with a group that was working with actually a cancer inhibitor, a heparinase inhibitor for cancer. This compound was made by Zucchero. And we were able to show that this molecule was a very potent inhibitor of attachment invasion of COVID, both in vitro in cell cultures and also in vivo in a mouse animal model. And most usefully, as time went on, we looked at it against the various emerging variants as they came along. And in fact, these molecules and heparan sulfate and heparin in general bound increasingly tightly to the emerging variants, giving an indication, I think, that the virus was actually evolving to bind more strongly to heparan sulfate on the cell surface as the pandemic went on, presumably because of the advantages that gave for infection. And it probably also altered the infection moving more into the upper airways as well, rather than being deeper in the lung. So these molecules could potentially be useful maybe as prophylactics in intranasal sprays or inhalers, and will also have therapeutic potential maybe for systemic treatment or airway delivery, and that requires further work. So they're very interesting. They have very high potency, and I think potentially could be used against not just COVID, but other viruses. They're already known to bind to things like dengue and respiratory syncytial virus. So we think there's an interesting opportunity maybe to keep studying these compounds as sort of first-line treatments against emerging pandemics of a range of different types of viruses in the future. And then a sort of a different topic, again, back to the nervous system, but generally to the central nervous system rather than just the brain, and particularly spinal injury and nerve repair. We collaborated for over a number of years with Sue Barnett at the University of Glasgow, studying nerve repair mechanisms. And in the early part of these studies, we were able to show that some of the cells they were testing for the transplantation, so you can transplant different types of cells into nerve damage to enhance the nerve repair. And one of those types of cells is Schwann cells, which are the accessory cells on nerves that wrap around the nerve. And those can be useful, but they tend to cause a scarring reaction when you put them into a site of injury. There's an alternative type of cell, the olfactory sheathing cells, which are found in the nasal cavity, up in the nasal cavity here, in the area where you actually sense smell. And these type of cells don't cause this scarring reaction. And it turned out the difference between them was that the olfactory sheathing cells express high levels of the 6-O-sulfatases that I mentioned earlier, whereas Schwann cells express low levels. And the difference then in the sulfation of heparan sulfate alters FGF signaling pathways, which are important for the scarring reactions. So that suggested the idea that maybe we could manipulate these molecules, manipulate heparan sulfate or use heparins of different types to control scarring reactions. And we went on then to study also systems related to multiple sclerosis and nerve repair, and we're able to show some interesting ability of some of the modified heparins, very low sulfated variants of these heparins, to promote remyelination, which is the problem with multiple sclerosis, is you lose the myelin sheaths around the cells. And you can promote remyelination with some of these compounds, suggesting they could be useful in vivo for studies on animal models. And indeed, we've gone on to, just in the last year, this was published just very recently in a couple of different preclinical models of animal models of multiple sclerosis. First, an inflammatory model could show very nicely that when you treat with the compound, you can lower the amount of disease burden and also alter the weight loss. You tend to get a lot of weight loss in these animals when they have this treatment. So you can see that here, and you reduce that when you give them a treatment with this heparin. So that's really exciting data. And then a second model, which is a model that focuses purely on remyelination, so it doesn't involve any kind of inflammatory responses. And in that model, we were also able to show significant improvements in these animals, more rapid return of myelination, less weight loss, etc. So we think this compound MHEP7 is a promising candidate for further in vivo studies, and we're now looking further at its efficacy and bioavailability. And then finally, over the years, we've looked at a number of potential cancer targets, and the modified heparins, we were able to show with a collaborator in Liverpool that low sulfated, low molecular weight chemically modified heparins are actually quite good at targeting a protein known to be important in metastasis of cancer cells, so the spread around the body. And this was a kind of Friday afternoon experiment because he had the cell assays and we had the compounds, and said, let's give it a try. We had no particular reason to think that there would be any effect here, but it did turn out to be very, very potent, and we were able to take that into animal models and show that with some of these compounds you could dramatically reduce the amount of tumour, this is tumour nodules on the lungs of these animals treated with different types of human cancer cells, for example melanoma cells. So they're promising leads as metastasis inhibitors, and we think they may also inhibit primary tumour formation as well, and that requires further studies. We also studied neuroblastoma. We had a very talented doctor who did a PhD in the lab, Val Solari, and studied neuroblastoma, which is one of the most common tumours in young children. And we were able to show that the enzyme SULF2, so the sulfatase, again popping up as being important, was a contributor to the oncogenic function of the MYC-N oncogene, which is the key oncogene which promotes this type of cancer, and in fact showed that if you down-regulated or silenced that SULF2 gene in tumour cells when you put them into mice, you could actually reduce the amount of tumour formation. And that's obviously very exciting because the sulfatases, as I mentioned earlier, are outside the cell, so they'd be easy to target with drugs, and we're currently looking at some inhibitors of sulfatases for that. And then I think finally we also looked at some of these mimetics of heparan sulfate, where you have these chemical arms with these sulfated sugars attached on the end of various types, and we were able to develop some of those that very nicely inhibit the heparanase, which cleaves heparan sulfate outside cells. So these glycomimetics proved to be very potent, and we tested those in collaboration with Israel Vodavsky in Israel, showed that you could dramatically reduce the amount of tumours in these mice, picked up by imaging here when you treat with this compound. So I think the take-home message then from this section really, where we kind of increasingly looked into the potential biomedical applications of some of the compounds we've developed and some of the systems we've studied, is that the heparan sulfates are indeed a rich territory for studying, for discovery of drugs that can target protein interactions of heparan sulfate, or indeed extracellular enzymes like heparanase and the sulfs, which are really important for different biological functions and in different disease indications. We have a number of different ways to make these compounds, so I think the future is very promising. We should be able to target these functions increasingly. We know we can target them in function. I've given you a whole bunch of examples for that. We can target them in vivo in animal models and show they work. And we've done that in multiple preclinical models, so Alzheimer's disease, nerve repair, COVID-19, and cancer. Just a whistle-stop tour through some of those that I've given you. So I think it's encouraging for the future development of novel therapeutic interventions, and there are certainly others looking at this, and there are compounds in development at the moment in clinical trials for cancer, and also for priming labour, where women are delayed in labour. There's a very simple treatment that can be given that's going into phase 3 trials at the moment. And I think if we get one or two of these to be successful, I think there'll be maybe a lot more interest in the field as being a very exciting opportunity to take heparins forward and to really show that you can teach an old dog new tricks. Heparin's been around for over 100 years, in clinical use for over 80 years, probably 90 years now. And it has some new tricks up its sleeve, I think, for the future. I don't know whether any of our things will go anywhere. It's really difficult to do drug discovery. It's very long-term and very expensive, but hopefully we've at least shown the way with some examples of compounds that could work and be part of the development of that story. And of course, looking back, research, there's always more to be done. I had this quote in my PhD thesis, and now I can look back because I am the other side of 60 and say, yes, indeed, that there is more urgency, there isn't as much time, but there's always more to be done. Research is always incomplete. We're all developing the next stage and standing on the shoulders of others. And of course, no man is an island. This is all dependent on other people, and as was alluded to before, I was definitely inspired by my biology and chemistry teachers when I did my A-levels at Calder High School in West Yorkshire. That was really important. I have to acknowledge the many PhD students, postdocs, and also technicians, and many different MSc, MRes, BSc summer students, et cetera, over the years that contributed to all these different projects, and the research is down to them as well as me. Also acknowledge collaborators for the first phase. So this is the people that I guess I particularly acknowledged back in 2005, at that time that I'd been collaborating with. But also the people in Birmingham that supported me. Ian Treyer was head of school, and then John Heath when I was there, and other colleagues, and they were really supportive at that early stage of my career development. So I really want to say a big thank you to them. And of course to the many people, I think I've listed most of the funding agencies that have funded my work over the years, and some of them still currently. And these are the ones particularly related to the work I was describing over the last few years on some of the in vivo studies on the compounds and different applications. And of course, the current group at Kiel that I connect in with, I have a part-time position at Copenhagen, and past workers at the University of Liverpool as well. And of course to Kiel for recognizing glycoscience and promoting it as an area to develop a center, so foresight there, and hopefully we're going to develop a really exciting center that's going to perform really well. And it's exciting to have the opportunity to be involved in that. And of course, I have to recognize the family team. So the Antipodean team, this was the pictures I showed back in 2005. My parents back in Australia. They're still alive. My dad's 90 next year, so they're still doing well. One of my brothers here, sadly, my youngest brother died of cancer about 10 years ago. But it's good to have them backing me. And then, of course, the home team, my wife, Marion, son, Matthew, and daughter, Sophie. This was pictures from back in 2005. Things have moved on, and of course Marion's family here, who are my UK family. The home team has gone somewhat global, so Matthew is now married to Courtney and is a Kiwi, is living in Auckland. This is them on honeymoon in the Philippines very recently. And Sophie and has married Ross, they're in Sheffield. They've now added to the family, I'm now a granddad as well. So this is little Felix, who's born in January. And of course Marion continues, and we've been married 40 years this year, so big thank you to them. And I'll just finish with this other quote that I saw from John Nash, which I thought was excellent. The most important discovery is the discovery that mysterious equations of love, that it's only in the mysterious equations of love that any logical reasons can be found. So there's more to life than research, basically. But it's been fun, and it's good to be here and to continue for a few more years yet. Thank you very much.